I think that certainly Nazneen's presentation highlights an area where we really have to start thinking about other clinical data and the quality of the clinical data that's being captured. We're talking very much at the forefront of the genetic information that's coming back, but unfortunately quite a lot of the clinical phenotypic data that is stored in our clinical notes is less than quality. And so with the value of these leading edge scientific discoveries that we're making is going to be with the linking of the genetic and the phenotypic data. And so I think we need to start thinking a little bit more about the quality of that other data that we capture as well. Anyway, let's go on to our final speaker for this morning. I think you'll all agree we've had some fantastic presentations this morning, and this one is going to be no less fantastic. So Julia Fan Li, and she's the Senior Vice President for Seven Bridges and is very passionate about working with developing countries. Julia.
Thank you.
Just quickly, for those of you who would like to come and take a seat, there's plenty of seats, and if those who have a seat could just shimmy along and accommodate, thank you.
Good morning, everyone. My name is Julia Fan Li, and I'm from Seven Bridges. I'm really excited to be here to share with you what we see as our future vision in the genomics industry. We see a few trends, and we'd like to outline and share those with you. But first, I'll start with a story. I first learned about the word cancer when I was 6 years old. I came home from school one day, in my first year of primary school, skipped home, and I found all these relatives in my house. Cousins, aunts and uncles I hadn't seen for a long time. And at first, I was delighted. They had all gathered in my home. But little did I know that while I was at school that day, my grandmother had passed away from leukemia. I don't really remember her being ill. I just remember eating watermelon with her. So in a small town in the outskirts of Shanghai, I can tell you my family did not know what subtype of leukemia she had, and nor did we have any access to tumor normal pair analysis. Today, we live in a completely different cancer world. We can use genomics to really drive the way we prevent, predict, treat cancers, and offer new therapies. We can see the level of somatic mutations and then compare them against background levels to see what significant genes are arising. And we're constantly adding to our repertoire of knowledge. So researchers have shown that as we sequence more and more cancer patients, we can find an increasing number of significant genes. This is a paper published by the Broad last year. And as we find more genes, we can see that these significant genes arise in a different number of frequencies, depending on the cancers and depending on the genes. We all know a handful of very famous genes that can be seen in the majority of cancers. And here we can see that as we sequence, in this paper, at least 5,000 or so patients, we can see the saturation of the genes that occur over 20% of the time. However, all cancers are different, and they're unique to the individual. What we really want to find out is the significant genes that are occurring at between intermediate frequencies of 2 and 20%. And for that analysis to happen, we really want to see the other lines on this graph, apart from the red line, also hitting saturation. This means we need to sequence more cancer patients to find out this more knowledge. The authors predict that if we had 100,000 cancer patients, we would be able to find out a lot more and tailor their treatments. A quiet revolution is happening. It's happening all around us. And at Seven Bridges, we believe that in the next couple of years, we are about to hit 1 million genomes being sequenced. This is now becoming a reality. We are involved in many projects that are over 100,000 genomes being sequenced. This was a huge number just a few years ago when announced by David Cameron. Genomics England is at the forefront of that. We have the Million Veterans Program in the United States and the PMI initiative. They have over 1 million consent forms from veterans who are willing to be added to a study. Sequence Bio is active in Canada in a province called Newfoundland and Labrador, where they also want to sequence 100,000 people. And all around the world, there are new initiatives launching all the time. 1 million is about to become real. As the next generation sequencing data explodes, we can see the subsequent data and the analysis will also explode. We will reach exabyte scale. This now puts genomics into a family of unlikely bedfellows, along with astronomers who love their stars, along with those kittens on YouTube, and along with the Justin Bieber followers on Twitter. Genomics have become one of the 4 major pillars of big data. We will need new tools to analyze this data. How can we efficiently handle all this information? What can we do better? At Seven Bridges, we see 3 major trends upcoming in this. The millions of genomes will shift how computation is done, how portable workflows are managed, and we also see advanced new data structures. First, we'll look at computation centers. As mentioned by some of the previous speakers, the Cancer Genome Atlas is a huge reservoir of knowledge for cancer researchers. It has been publicly funded by the National Cancer Institute in the US as part of the NIH. And currently, in order for you to research this data, you need to download the Cancer Genome Atlas onto your local supercomputers and servers. So this means each research center needs to build their own high-performance cluster in their backyard, download the data before they can even ask it a simple question. This might look easy, as if we were downloading an Apple iTunes app, but unfortunately, 2.5 petabytes of data is more difficult to download. Each of these research centers will need a 10 gigabit uninterrupted connection and over 23 days to download the data set. If they do it wrong, the whole process has to be repeated again because you only know you've done it wrong once it's all downloaded. So in speaking to researchers, this is a huge bottleneck. They cannot ask the questions they want in the silos that they work in. We see that in the future, and future meaning now, that the data can be stored in a cancer cloud. We are part of a pilot initiative that are putting 2.5 petabytes of this NCI data, the TCGA data, onto the cloud. This is now live at cancergenomicscloud.org. We've put the data in the cloud, and we've added appropriate access controls and audit logs to see what is being done with the data. This means that the original research centers in the previous picture no longer have to download the data or build capacity to download the data. They can bring their tools and workflows to the data, ask the questions, and then get back the answers in a more timely manner. So this means we think that data repositories will be replaced by networked computational centers. This will help collaboration. So if you imagine a scientist who had contributed to the TCGA data themselves, in breast cancer, for example, they in the past really had only full knowledge of that breast cancer set data. But now at the cloud, that data is collated with all the other cancers, and they can ask significantly different scientific questions. They can ask more questions, and if they get the right answers, they can move on that. And maybe even more importantly, if they get the wrong answers, they can fail quickly and pursue new different research paths. This will also help reproducibility because each of the questions that you're asking for the data set is also tracked so that other researchers around the world can replicate what you're doing. This leads me to the second trend we see, which is that portable workflows will also help data transfer and reproducibility. In many science papers, we see that everybody is clamoring for information. When landmark papers come out, everybody wants to know how it's done and be able to reproduce the data themselves. However, the current way we share analysis sometimes is the abstract, the introduction, and the method section stuck at the back may or may not have even a pipeline picture. We don't know exactly the steps that are done in this analysis, especially when the worlds of bio and IT come together. In the TCGA dataset alone, there are over 10,000 tools that are being used on these datasets. These bioinformatic tools are constantly improving, sometimes every hour and sometimes in weeks. When new versions are made, it's very hard to remember everything that you did to improve upon the previous version. This needs to be tracked somewhere. And in most papers, researchers use over 50 tools alone. We predict that in the future, when research is published, there will be a link to the exact pipeline that was used to produce the analysis. These links will be able to be shared, uploaded, copy and pasted to other researchers. This is not tied to a particular service or a company, and you can share this across different platforms. We think that this will help the data that's being produced in scientific papers. It will be able to be shared, and it will allow the data to be trusted. What we all hope for in research is that our data is proving something, being trusted, and ultimately the clinicians will trust the data, tell that information to patients, and the general public will also understand the research. One way that we've implemented this objective of portability is the common workflow language being developed at Seven Bridges. It's not just us who are asking for this common workflow language to be adopted. It is ourselves with other ecosystem partners in the bioinformatic world. We're working with other companies, open source projects, and protocols to find a common way to describe the tools, data, pipeline, and parameters we all use. We currently deploy the common workflow language as Rabix. You're welcome to contribute to this effort at GitHub. We also host bi-weekly meetups online, and please contribute if you would like. We implement this common workflow language on our systems through Rabix, and you can query our data using this language, at which point you can also then share with your colleagues, and they will know exactly what you're speaking about and which tools you used. So these open standards, we believe, will improve reproducibility and accountability for the science that is being produced. Rabix.org is the website, and you're welcome to contribute. A third trend we see is the advanced data structures. This will happen now because we have the data sets that enable new tools to be used at population levels. An example of this is when we each want to think about what our genomes look like towards the reference genome. How do we compare to what's defined as a healthy human? When you compare your genome, or when I compare mine, that is currently being used to a reference genome that is linear and static. What we predict in the future is that as more and more molecular information is generated, there are new self-improving algorithms that can learn from all of our data. And as it learns, it can improve the accuracy of which it calls variants on your particular genome and on my particular genome. I can illustrate with an example. So if the 10 of us had our genomes sequenced, we would have the sequence outputs. And currently, it is being compared to a linear reference to call the variants. Now, in a linear reference, it cannot remember each of our individual differences. It can only remember our change to the delta that is the constant linear reference. In the future, we think that this linear reference can become dynamic. A way to do that is by using what's called graph genome, a self-learning tool. And as each of our 10 genomes is sequenced, we have different variances in our genomes. A graph tool, or a dynamic graph of a reference genome, can remember the differences. It can see and remember the frequencies of which it sees the variations and assign values. In the future, the graph reference can really be helpful at population levels when we feed it more and more genomes over time. For example, Genomics England is currently working towards a population level dynamic reference genome, so that in the future it can more accurately call British-specific human variances in their population. So we can represent these genetic paths with metadata and learn from ourselves. So the potential of graph genomes is very large. It has no bias, meaning that we can add new variations over time, and it doesn't just have to be for the human genomes. It is an elegant way of a self-improving machine learning tool. As you give it more data, it will learn and remember. It can also be tailored for specific targeted references. So for example, we can adapt the same tool to be a cancer graph genome, for example, and we can then drill down further to different types of cancers as well. There are two additional competitive advantages for having a self-learning genome reference rather than a static genome reference. One is compression. Instead of storing every single linear genome, you can store just the frequencies of the variations. And this makes it easier for computational analysis. By using the data of the 1000 Genomes project, we can store all the 81 million variants in just 20 gigs of data. This will help the downstream compute resources that are required to do research. And second, the graph genome is completely anonymous. It doesn't remember each individual genomes as we feed it. It only remembers the frequencies that it sees and that it learns. So if I were to go into the graph genome, I can't go in and unpick all the different people that contributed to it, but I know that it's a very accurate representation of all of us. So is this just a nice theory? Why haven't we seen this before? The Seven Bridges has a very strong research and development team, and these are some of the early results that they have shared with us, and I'm here to share with you. This is our alignment tool of the graph aligner compared to some of the most commonly used aligners in genomics. As a breakdown, we can see on the y-axis the sensitivity, or how accurate the variant calling is. And then on the x-axis, we can see the SNPs and the indels at different deletions and insertion base pair levels. We can see that at the SNP level and in short reads of under 150 base pairs, all of the aligners perform quite well. But the graph aligner in red performs well over longer reads and in both deletions and insertions. So to break down this graph, we've compared it straight to BWA, a common tool. Again, performing very well. GATK plus BWA performs better as a tool, but also not as well as graph. And we've added some more common tools to the mix as well. We want to continuously improve this tool over time, and we are working towards a launch of this tool as well. If you would like to join us in trying out this tool, please sign up as a beta user at sbgenomics.co.uk/graph-test. We're accepting users now to test this tool, and we are looking for a commercial launch this year. So we see graph genomes as an example of an advanced data structure that will replace the flat files. It is constantly improving, and it can really scale to the millions of genomes that we think the world is moving towards. It can really help population-level genetics as well. So in summary, we see the three trends as the computation centers change into network structures, the portable workflows that allow us to bring our tools to the data rather than the data to the tools. And then we also see advanced data structures, like graph genome, that will help analyze larger and larger data sets using new tools that we have never used before. Maybe at this point it's important to point out how a company like Seven Bridges, that bridges both the biology and the IT, can contribute to the larger genomics ecosystem. And tying in our work to the previous speakers, we know that Alex talked about national scale projects with Illumina. As these nations see the value in precision medicine, they will need tools that build population-level genomes for themselves. Genomics England has taken a lead in this, but we see this technology evolving to other populations and countries as well. Second to Mene's issues at AstraZeneca and connecting research in precision medicine, we see portable workflows as a great enabler of pharmaceutical research. So in the way that multinational pharmas are structured, there are research centers all over the world, in Cambridge, in Boston, in Singapore. They need all of their bioinformaticians to work together and know what each other are doing. They need to have secure, collaborative spaces. And one way to do this is having a cloud structure, where if you go onto the cloud scientists can make changes in real time, and then each change is tracked and versioned so that when you wake up the next day and your Singapore scientist has made a change, you know what you're working on in London. And when you sign off at work, your Boston colleague knows what you've done as well. And then to Professor Raman's comment about clinical interpretation, we see that graph genome and these new tools can be helpful because as researchers communicate what we find to clinicians, they also need to have easy ways of looking into the black box of bioinformatics and seeing exactly what we're doing. We need to make sure that communication lines are open and that the doctors are interested in using the discoveries that we find in research. Ultimately, this will help patients then also learn and trust the doctors who trust the researchers. So I've lost 3 grandparents to 3 different types of cancers, and I have 1 grandmother left. And my wish in the future is for my grandmother and other grandparents and other families of loved ones to be able to use the biomedical innovations we see in genomics in discovery, but not limit to discovery as it goes downstream and is translated into products and tools that we can use for patients. There's a long way to go, but we thank you from all of the global teams at Seven Bridges and look forward to meeting you later today.
Julia, I think we have time for just one question, if there is anyone. There's a hand gone shooting up there for you.
Hi, thanks very much for a very interesting presentation. I have a question regarding the graph genomes, because being a genomicist myself, I've been obviously working quite a lot with sequential references. And it looks like there are lots of advantages for using graph genomes. So my question is, what are the disadvantages?
Well, the primary disadvantage is that we need to prove the tool on real data sets. And the only way that the tool will improve is if we are able to make improvements on these data sets. The graph genome itself is not a new discovery that Seven Bridges came up with in our 6 years of history. The graph genome was first published in theoretical literature by mathematicians in the 1980s. But this tool just wasn't developed or commercialized because there wasn't the data sets large enough for it to learn from. So as we get from 1,000 genome projects to 100,000 genome projects with Genomics England, we need to improve the tool and prove that it works on a population level. We do this in collaboration with scientists and researchers to begin with, and also with national scale government projects. So we hope that is the one current disadvantage of the tool, but as with all new tools, we all seek to have collaborations with early users and adopters to prove it.
Thank you. And I think I'd like to thank you as an audience. You've been brilliant, but I think we really must give another round of thanks to all of our 4 presenters this morning. They've been really inspirational. So a big round of thanks.